So bacteria cells can multiply by simple cell division, and you can see one cell can split into two, and then those can then split into two further, and so on and so forth. You can see how bacteria growth can explode very quickly, and this type of growth or cell division is what we call binary fission, and this is how bacteria cells grow. And essentially this can happen as often as every 20 minutes. As long as two things are fulfilled, and those two things are, it needs to have enough nutrients, the bacteria, and the temperature needs to be correct as well. And if those two things are correct, then as often as every 20 minutes, binary fission can occur, and you can see how quickly the bacteria can grow. Now, we can actually choose to culture these bacteria ourselves, and there's two main techniques that we can use. So we can use either a nutrient broth solution, and essentially in this case, what you have is a broth liquid that is full of nutrients, and this allows for the bacteria to grow inside of it. Okay, so the bacteria is grown inside of solution. The other way we can culture bacteria generally is through colonies on an agar gel plate. And essentially what agar gel is it's this very nutrient-rich gel, which provides all the nutrients needed for the bacteria to grow. And essentially you take this agar gel plate, you put it in, might use a petri dish, and you then put it into an incubator to keep the temperature there at a certain temperature and allow there for the bacteria to grow. We can say you grow on a petri dish. So it's not in a solution this time. It grows as different colonies of bacteria on a petri dish. We'll look at that in more detail. And essentially what we do need to know is that we do want uncontaminated cultures of microorganisms or bacteria if we want to investigate disinfectants or antibiotics. So this is really important. Whenever we want to do kind of investigations to see how effective a disinfectant or antibiotic is, we need uncontaminated cultures. And that's why in the first place we're interested in growing these bacteria. So then we need to look at how do we actually prepare an uncontaminated culture. And we do this using what we call an aseptic technique. And so what we might do is use an inoculating loop. So an inoculating loop looks something like this. It's basically just a loop of metal, and the idea is you can dip this loop into some bacteria and cover the loop in that bacteria and use that to transfer different microorganisms. Before we do that, we want to actually sterilize, and the way we do this is we pass it through a flame. Okay, and so what that might look like is some kind of Bunsen burner over here. You have your flame, you put your Bunsen burner on, and you basically make sure you put your inoculating loop into this flame, put it at a very high temperature. This kills all the bacteria and essentially ensures that we have a sterile inoculating loop. That means when you put your inoculating loop into your actual bacteria, so you might have a petri dish over here, this actually has the bacteria that you want, and you take your inoculating loop once it's sterilized, you put it into your petri dish, you can then kind of swab it around, you take your inoculating loop out, it's now got that bacteria you want, and you can put it into a separate dish to start to grow it. And so for example, you could then transfer this to an agar plate. Okay, so a plate filled with agar gel. And then if you want to cultivate this, then what we do is we might put this into an incubator. And the idea of an incubator is to keep the temperature at a good temperature for the microorganisms to grow. And generally in a school lab, this tends to be at 25 degrees Celsius. Okay, we don't want it any higher than that because bacteria that grows in humans tends to thrive at human body temperatures, which is around 35 to 37 degrees Celsius. So if you put it much higher, then we're going to end up growing bacteria that could actually grow inside of our cells, which we don't really want. So for safety reasons, we keep it at about 25 degrees Celsius to encourage bacteria that grow at these kind of temperatures. Okay, so that is basically how we might grow a bacteria culture. And let's look at a few examples of kind of practical questions you might get in an exam. Okay, a few questions here. Feel free to pause the video and have a go as usual, and put your answers in the comments below. So question 1, I start off with 10 bacteria cells. The mean division time for this population is 35 minutes. How many bacteria cells will there be after 2 hours? So let's work this out over here. So we've got 2 hours and we want to know how many divisions are going to take place. So if the mean division time is 30 minutes, let's convert this both to minutes. So 2 hours is equal to 120 minutes. Divide that through by the 35. And if you put that into a calculator, you should get 3 divisions. Okay. It will be 3 point something, but the idea is you don't quite reach 4 divisions, so you only get 3 divisions, okay? And we're using the mean time for the population to divide. So if you get 3 divisions and we start off with 10 bacterial cells, then we know that every division binary fission occurs, so the number of bacterial cells will double. So if you start off with 10, then essentially this is going to double, and it's going to happen 3 times, so it's going to be 10 to the power 2 to the power of 3. Or in other words, 10 times 2 for the first division, times 2 again for the next division, and times 2 again for the third division, which is the same as saying 10 times 2 cubed. And so 2 cubed is equal to 8. And so 10 times 8 will give you 80 bacterial cells. Okay, so hopefully everyone got that one correct. Question number 2, what temperature should microorganisms be incubated at in a school laboratory? We'll just write this one up here. So you should know in a school lab, you're going to use 25 degrees Celsius. Okay. Question number 3, how would you sterilize an inoculating loop? To sterilize an inoculating loop, you want to hold it over an open flame. Okay. So pass it over an open flame, and the heat from that flame will basically kill off all the bacteria on that loop and make it sterile. So hopefully you got this correct, and I'll see you guys in the next one.